first of all, you know, if Friday is a good day, if Friday's a good day at noon, I'll meet out here for anyone who wants to meet out here for lunch. I know this is just for hanging out purposes, not for study purposes. Unless you want to ask questions, but you'll just have had the exam, so I don't know that you necessarily want to ask questions. But you're welcome to ask questions. But just hanging out kind of day if you want. If it's a nice day, anyone who wants to show up, show up. Since only 7 people showed up for the review, I don't know how many people will do it. No, it's more than 7. It's about at least 12, right?
Yeah.
It wasn't very many.
I was bad about turning the camera during questions, but,
okay, but
I was
did
tired.
get a lot of the stuff
Yeah, pretty much all of it.
Okay, good.
Like, it's just a little. You can hear it.
Okay.
Put in headphones, it's fine.
I don't usually look at these things.
Well, no.
I like to see myself on these things. Anyway, but look, so the test is Wednesday. It's going to go through, here I should put this, through fermentation, and it's just the beginning of today's lecture, in other words. Beginning of today's lecture. And I just want to clear up a few things I didn't quite get to. I did a little more in this section than I did in the next lecture section, but still, I just want to make sure it makes sense to you. So we've already done glycolysis, and we actually started fermentation, but I just want to go quickly talk about what these things mean, so that it all does make sense. Glycolysis, by the way, let me just put these things down.
Breaking down sugars.
Glycolysis is, means the breaking down of sugars. This is something I mentioned to you that most every organism on Earth can do. It's something that most every organism does. And what it is, it's you take glucose, which is a 6-carbon. I'm just going to put C for carbon. It's a 6-carbon sugar. 6-carbon sugar. Okay. And in a series of steps, and we're not worrying about what the steps are, you end up with 2 pyruvic acids. These are 2 carbon molecules.
So it breaks
3
down.
carbon molecules. Sorry, what was that?
It breaks glucose down.
I'm sorry, say it again.
It breaks glucose into those.
Glucose into 2 pyruvic acids, and each pyruvic acid is 3 carbons. Okay, so we haven't lost any carbon. No carbon dioxide or anything else that happened. But 2 pyruvic acids are produced, and 2 ATP molecules are produced. Remember, this is what we're trying to do. If you want to talk about what we're trying to do and what we're trying to accomplish is make ATP. Why do we want to make ATP?
For energy.
I call it that dollar bill of the energy system. In a catabolic reaction where you're breaking things down, you would like to trap some of the energy
in the high
by
energy
making ATP. Then when you need energy to run some other reaction, you use ATP to force those other reactions to go, anabolic type reactions. Okay, that makes sense?
Yep.
So the whole idea here with this glycolysis and fermentation and cellular respiration is to make ATP so the cell can have the energy to do whatever it needs to do. All right. And by the way, 2 ATP is not very much ATP, not very many ATP, I should say, for one sugar molecule, because the sugar molecule has a lot of stored energy in terms of chemical bonds. It's a little, but not very much. Now, this takes place in the cytoplasm. So this takes place in the cytoplasm, okay?
Okay.
And it's also anaerobic. And what do we mean by anaerobic?
No water.
I mean, it says no air, without air, but really we're talking about oxygen. So no oxygen is involved. No O2 used. All right. Now, I said for reasons that I wouldn't bother worrying about, you can't stop at glycolysis. You can't stop and say, okay, pyruvic acid and a waste product, we're done with the reaction. No. Something has to happen to pyruvic acid. And and the 2 possibilities are either fermentation. Well, actually, there's another possibility that I'm not going to talk about. But for us, 2 possibilities: fermentation or cellular respiration. So we'll start with the fermentation because that's what we're going to do today. So fermentation. And if I put plural on there, it's just to tell you there are many different fermentation pathways. I'm going to give you two as an example, but there are hundreds of different possible fermentation pathways. Let me go back a second and say one more thing here. I did say it, I want to say this again, the glycolysis. I said that most organisms on Earth do this. Most organisms do this. All right, so this and I'm kind of distinguishing that from who can do fermentation versus who can do respiration, right? Fermentation. These are done mainly done by bacteria. What kind of organism are bacteria? In terms of two main cell types, prokaryotic cells. So this is mainly done by bacteria, but some eukaryotic cells can do it. Some eukaryotic cells can do it. If there's not enough oxygen around. I'll just say isn't any O2 around. But not enough. You could say not enough O2 is around. The example is yeast. You know, we use yeast to make bread or to make wine or something like that. Yeasts are fungus, so they are eukaryotic, so they can do cellular respiration. But if there's not enough oxygen, they'll ferment. Okay? Now, what's the deal on let me tell you what the overall idea on fermentation is. And I did have this on the board last time, so this isn't new information, but still I want to make it all make sense to you. So fermentation is mainly done by bacteria. And what happens is this, that after glycolysis, let's just say, start with glycolysis, all right? So we'll start with glycolysis. And we'll see that when we talk about cellular respiration, we'll also see that we'll start with glycolysis. We don't have to, though. We can start with fats. We don't go through glycolysis with that. I'll get to that. But anyway, so fermentation, we start with glycolysis, and the final products. Let's see. These are the final products of glycolysis, pyruvic acid and ATP. We're not going to talk about the ATP other than, hey, we're storing it. We're trying to save it up. We're in the bank. So think about these pyruvic acids. You think about it as a waste product. But something has to happen with that pyruvic acid. And that's what's going to happen. The fermentation is going to take the pyruvic acid, whatever the kind of fermentation is, and convert it into something else. But in those final products, there will be some organic molecules as waste products. No, that's not a, see,
Organic.
you may or may not have carbon dioxide as a waste product. Carbon dioxide is not
organic,
right? Even though it has carbon, it's not organic. Let me write it down and then we'll get your question, okay? So start with glycolysis, and the final products are some organic molecules. No, hang on, just wait, we'll see it. Organic molecules. Now you say, well, this is an organic molecule. Absolutely, pyruvic acid is an organic molecule, but that's not the final product. It's the final product of glycolysis, but you can't stop there for reasons that I'm not getting into. You have to do something with that glucose, those pyruvic acids. Yeah. Now wait, let me get your question before I go on.
The ATP just transfer down.
Yeah, I mean, just when you see the ATP, just say, hey, I'm just putting that in the bank. Okay. So I'm not gonna. But let's just continue. To start with glycolysis and final product are some organic molecules, but no more ATP are made than the two already made glycolysis. Hang on, just one second.
Oh, crap, what was that?
Okay, so the point is that with the fermentation, really what that's doing, you remember I said that we can't stop at pyruvic acid. So really what these different fermentations are doing is saying, hey, let's just do something with that pyruvic acid so that whatever it is, we can have it as a waste product and we're done. Okay? And so the two examples I gave you, and there, as I said, there are really dozens and dozens of different fermentations, but the two that I give you are the two
lactic and
common, and you know about them, and you know about them. So here, these will be the
alcoholic.
two examples of fermentation. And keep in mind there are many others. These are just very common because we use them all the time. One would be the pyruvic acid. Now, pyruvic acid this is for each pyruvic acid. I mean, remember there are two of them from the original sugar.
Yeah.
Each pyruvic acid is what's I'm talking about here. Each pyruvic acid is converted into an alcohol plus a CO2. And the alcohol is a 2-carbon alcohol. It's ethyl alcohol or ethanol. In fact, you see ethanol because now they're putting in some of the gasolines, right? That's the ethanol that we're talking about. That's the alcohol we're talking about. That's also the drinking kind of alcohol. Don't drink the gasoline with ethanol, please. That would not do wonders for your body. Yeah.
I was just going to say, so that breaks off one of the carbons with oxygen.
Yes.
And
Yeah.
then
So
leaves
one
the
of
two
carbons is
remaining
removed
2-carbon
by CO2,
alcohol.
And the other carbon of the these 3 carbons, the 3 carbon molecules. So of those 3 carbons, one of them comes off as CO2, and the other comes off as the alcohol. Now, and I said this to you last time. You know, when you're making wine or beer, what you want is the alcohol. Okay? When you're making bread, what you want is the CO2, because the CO2 makes the bread rise.
Yeah.
And you know there is alcohol in that bread before you bake it. As soon as you bake
evaporates.
It's gone. And then if you do any fermenting, you know, at home, if you want to take some apple juice and put some yeast into it, you'll start getting bubbling. And the bubbling, of course, is the CO2. And so what's happening, of course, is that the glycolysis starts at, you know, you're taking whatever the sugar is, glucose, and it's being broken down eventually to CO2 and alcohol, right? So remember, this whole glycolysis starts first, and then we take the pyruvic acid and end up with this. So this is the waste product. So for that yeast, some bacteria can do this exact same thing, but the yeast, alcohol is a waste product. You can't do anything with it. We can drink the alcohol and we can get some energy out of it. Okay, it's still an organic molecule. That is an organic molecule, right? Some organic molecules are the final products. CO2 is not organic; that's inorganic, right? But alcohol is. So in other words, we could drink the alcohol, we get energy from it. So that's a type of alcoholic fermentation, that's what we call it. But I don't care whether you use that term. I just want you to realize that this one example of fermentation would be convert the pyruvic acid to alcohol and CO2. The other example I told you about is taking pyruvic acid
and making lactic acid.
And again, that's 3 carbon, and it gets converted to lactic acid. It's also 3 carbon, so notice that there are no CO2s given off in this fermentation. And lactic acid is an organic molecule just the way pyruvic acid is. They only differ by a couple of hydrogens, actually, but we're not going to construct formulas. But anyway, but lactic acid is the waste product. We use this type of fermentation to make yogurt and cheese and various things like that. We also use, and it's a bacterium that does it. It's lactic acid bacterium. Lactobacillus is what it's called. We also, remember I said that some I said that, where I say start with it, there it is. But some eukaryotic cells can do certain fermentation, some fermentations if there's enough oxygen around. And this is the fermentation that our muscles can do if we do strenuous exercise. And we start building up lactic acid. In other words, you're trying to make ATP faster than you can make it aerobically, and you do a whole bunch of this fermentation, then you start building up lactic acid. So the way to make ATP very fast, as it turns out, when you're strenuously exercising, but you can't keep it up. You only do it for a short amount of time because we're not anaerobic organisms, we're aerobic. We have to stop and rest and then kick this lactic acid back to pyruvic acid and do our normal respiration. But anyways, but this is something that I mean, what if our muscle cells, when we're strenuously exercising, did this kind of fermentation instead? We'd probably have people out exercising a lot more, don't you? Whatever. Yeah.
Isn't lactic acid what makes your muscles really
Tired, yes, tired and sore and stuff like that. Yeah, that's right. And that's why you have to stop and take care of that lactic acid. And if you ate lactic acid, actually you do get lactic acid if you eat, cheese and
milk.
You get some lactic acid. Nothing wrong with that. It's an organic molecule. We can break it down and get energy out of it. Okay, and we do.
Mm-hmm.
So anyway, so that's really that's where the exam is going to go right through that. I want you to understand what a fermentation is. It's anaerobic. Did I put anaerobic up here?
Yep, you did, but you erased it. Well, no, it's right there in red. Anaerobic.
Okay,
no,
well, that's
no,
because
it's
this I'll just say this is still remains anaerobic, and there's no. I'll just anaerobic. Just make sure that it's an anaerobic process, and that's really so totally different from cellular respiration, which uses oxygen, and you'll see that we get so much more ATP out of it. Let me just give you a hint, and we'll come back to it. But in a fermentation, what's the maximum amount of ATP you get per one sugar? How much? 2 ATPs. When we get to the cellular respiration in a minute, you'll see that we'll get a total of 36 ATP per sugar.
Oh,
so respiration is so much better in terms of making ATPs. Steve. What?
If it's anaerobic, when you break it into alcohol, where does the O come from in the CO?
No, but there are oxygen molecules on the sugar molecule.
Okay.
And on the pyruvic acid.
Okay.
Yeah, this didn't come out of the air. This was
I was like,
because all the sugar, the sugar molecules. I mean, if you looked at it, you'd see a carbon with an OH and an H, an H, and another carbon with an OH like this. So there's all these OHs I mean, oxygens all involved in that molecule.
Okay.
So you're getting it from this, not from the air.
Okay.
But in respiration, you need oxygen from the air.
Yeah.
Breathing, right?
Yep.
Okay, so does that make sense? Now let me just leave this for the minute. I'm going to have to erase it so I can use more of the board. But let me just leave this for a minute so I can talk about what happens with cellular respiration. Cellular respiration, you can also call it aerobic respiration. Same thing. So we'll put this as the other alternative.
Oh,
cellular respiration.
Gotcha.
It's also called aerobic respiration. Same thing, okay? It means the exact same thing. And I mentioned to you, it's one of my little things because I studied bacteria and bacterial metabolism, that I get really upset when somebody in a textbook, calls fermentation or glycolysis anaerobic respiration. I go, I told you about this already. And note in your lab for this week, do your labs tomorrow and Thursday, it's on cellular respiration, but they do have a thing about fermentation. You're going to do an experiment with fermentation. It's kind of a fun experiment, actually. You get to design your own experiment. But unfortunately, it was miscalled anaerobic respiration. So I made a big deal about my students in my lab crossing the word anaerobic respiration and replacing it with the word fermentation. It is a fermentation. It's not anaerobic respiration. There is such a thing as anaerobic respiration, but only certain bacteria do. But what about cellular respiration? Who does that?
Plants.
Okay, it's done by all eukaryotic cells and some bacteria. There are some aerobic bacteria that do it. Okay, so cellular respiration is, I'm going to put in green because I had the green up there, done by all eukaryotic cells.
Chloroplast.
And some bacteria. Okay. So some bacteria can do it. And in the process, it's going to start, well, it can start with glycolysis. It doesn't have to start with glycolysis. For the bacteria to do it, has to start with glycolysis. For plants, it has to start with glycolysis. I think. But for animals, it can start with fatty acid metabolism. I'll try to get to that maybe today, I hope. But so it's not. But I'll say. So the point I'm going to say is that it can. Well, I'm going to say it starts with. I'll just say starts with glycolysis. And then I'll put in parentheses for fatty acid metabolism. But we'll come back to the fatty acid metabolism. So don't worry about this part right now. Just ignore this for a moment. Have it in your notes, but ignore it for a moment. Okay. So it starts with the glycolysis. So what I want to do is just keep it short for now, and then we'll get in more detail. So notice here, fermentation starts with glycolysis. What about the final end products of fermentation? Some organic molecules are there, but the final products of cellular respiration are totally inorganic, CO2 and water. Okay, so you're totally breaking down a sugar into CO2 and water, nothing organic. And that's as broken down as you can get. That's as oxidized as you can get a carbon, a CO2. So it starts with the glycolysis, and the final products are CO2 and water. Okay. And 36 total ATP made. I mean, that's so much more than the 2 you get if you just ferment it. Okay. 36 total ATP made. That's 2 from glycolysis.
And 36 from respiration.
34 from respiration. So what I'm trying to do right just at the moment, without getting into any detail, is just getting you to see the contrast with fermentation. You don't need to know this part for this exam, the cellular respiration part, which is all the other stuff for this exam on Wednesday. But now that we're covering this, I want to make sure you see the contrast. And I think you can see why this is such a big deal. It's such a big deal because look at how many ATP we can make. By the way, when I was a student way back when, they said 38 ATP. And then several years later, they said 36. And I just said to myself, can't people count? But actually, it's not that simple. What it is, when they're doing these kinds of experiments, these are tricky things to do to be able to actually get the total numbers for whatever's happening. And this is ideal conditions, blah, blah, blah. And there's a reason why they thought it was 38, and now they know it's 36, but I'm not going to worry about that with you guys. But anyway, just the main point here is that why is it such an important thing for eukaryotic cells to do?
Because that's how they function.
You get so many ATP. And look at this. We're big multicellular organisms that can do all kinds of things, send people to the moon and all that.
It's just way more efficient.
And look at look at bacteria. They're just these little one-cell organisms doing their thing, Now, I said that, didn't I? And yet I told you in my micro class, I say bacteria rule, because obviously we can't be on this planet without the bacteria because of all the cycles of nature and everything else. So I don't want you to think just because you only can ferment that you're some lowly critter. No. On the other hand, if you can only ferment, you're not likely to make rocket ships and send them. I know that I'm being speciesist, and I told you, but I am glad I'm a human. I really am.
No, I'm biased species.
I don't want you to think that I think that I'm so much better than all the other organisms on Earth. I'm one part of all life on Earth. Yes.
Do our bodies or other eukaryotic cells want to make as much ATP as possible, or do we stop after a certain point?
Well, it's a good question. I mean, do they want to? I mean, I like the word want because I use the same terms, But no, it's a good question. And what happens is there is regulation. You don't just keep on building up ATP and ATP. if people that are, stock traders or whatever, somebody, they keep on building up dollar bills and the bank account gets bigger and bigger. But no, in your body, or in individual cells, you will make ATP as you need it. And to make ATP, I did give you this thing before, I think. ADP plus a phosphate is how you make ATP. And then when you use up the ATP for energy, but this takes energy, you have to put energy into the system to make it.
Mm.
But when you take ATP and use it up, you get ADP plus phosphate, and energy is given off. That's just a general pattern. But here's the deal. Your cells maintain pools of all kinds of things. You have pools of amino acids and pools of nucleotides and pools of ADP and pools of ATP. Your cell maintains pools of all these various things that it's going to need. And in the case of ATP and ADP, you have some ratio of ADP and ATP. And let's just say that this is ADP. As you start using up ATP, that'll go down, and the ADP goes up. That'll kick off all these processes, the glycolysis and the cellular respiration to go. So you remake the ATP, so you have enough for whatever you need. So it is regulated. It's a very good question, but it is regulated, and that's a neat thing to think about. Yeah.
Isn't the ATP stored as glycogen?
No. That's one way we store sugar to use for the future, but that's we'll worry about that in physiology, not in this class, because that's the liver and
I
everything.
know, but like when you say, like she was saying, it makes.
Well, I mean, when after a meal, okay, we have more energy available than we're going to use right now, so we will store a certain amount of that as fat, a certain amount as what's called glycogen, which is really starch. It's the same kind of thing as starch, but animal starch. And that just stores the extra for use later. Yeah. But that's not ATP being stored. It's you're really storing sugar molecules.
So you can
Which when we need them, we'll break them down in these
yeah.
processes to get all the ATP we need. Okay. All right. So you see the difference here. This is a big deal. This is a really big deal. Is it getting warm here, or am I having hot flashes? Is it getting warm?
Yeah.
Yeah, it's the sun. It's coming out.
I'm not just saying the door keeping it open. But anyway.
No, you're right. No.
It is getting warm. Okay, so now let's talk about cellular respiration. Now in this case we're going to start with glycolysis. Okay. And what do we have at the end of glycolysis? 2 pyruvic acids. Where does glycolysis take place? In the cytoplasm. Where cellular respiration takes place in eukaryotic cells, the mitochondria. Okay?
Oh.
So done by all eukaryotic cells. Let me put this in the mitochondria.
Important to know.
Now, if you're a bacterium, you don't have mitochondria. In fact, I'll tell you about the origin of mitochondria.
I mean,
don't worry about that. Just worry about the eukaryotic cells. It's down in the mitochondria. And cells have hundreds of mitochondria in them. Something like a liver cell may have 1,000 or more mitochondria. A typical skin cell might have several hundred mitochondria. So these are common. Muscle cells have hundreds and hundreds and hundreds of mitochondria. So these are important organelles. Remember, one of the many membrane-bound organelles. Actually, 2 membranes.
do we have chloroplasts too?
No, we don't have chloroplasts.
We have okay. We have
Plants have
mitochondria. We have mitochondria. That's what I was checking.
But all plants have the mitochondria too. All eukaryotic cells have mitochondria.
Mitochondria. Okay, that's
Only
what I wanted to know.
Some of them have chloroplasts, like plants and algae and all those. But anyway.
Okay.
But so one of the terms that people use to remember what the mitochondria are, that they're the powerhouse of the cell. I love those types of terms. But they are the powerhouse in the sense that the majority of the ATP, the majority of the ATP that your cells make, come from cellular respiration, which occurs in the mitochondria. We do make some out of the cytoplasm. We make some, a little bit, but
So
small
the mighty
amount of
mitochondria.
ATP out of the cytoplasm compared to the huge number of ATP molecules we make in our mitochondria. Right? Does that make sense so far?
Yeah.
Okay, so we're going to do respiration now. And we divide it into two parts, but keep in mind that those two parts are intimately associated with each other. They don't do one, you do one and then you do the other. No, they're constantly happening together. But let me tell you about it. So, first of all, one other thing that I didn't put up here, but let's do, which to contrast it with glycolysis and fermentation, is that respiration is aerobic, meaning that oxygen is required, O2 required. Without O2, respiration would come to a glaring halt, grinding halt. Sorry, that would be grinding halt. All right, well, what's the deal in respiration? Well, those pyruvic acids diffuse into the mitochondria. Right? So this is here. I kind of had this picture, I think, before. So if we say that this is cytoplasm up here, and down here is mitochondria, does that make sense what I'm saying with my line? Then these pyruvic acids diffuse in. Okay? They just diffuse it. It's just diffusion. You start producing all these pyruvic acids, they start going into the mitochondria. By the way, the singular of mitochondria is mitochondrion, I-O-N. You don't see it in the singular very often. Almost everyone says it plural mitochondria. And you have so many of you might as well. And whenever we talk about a reaction, remember this reaction is occurring millions and millions of times in like the mitochondria. The glycolysis is millions of times. It's not just one reaction, it's a whole bunch of them. This is happening over and over again. Anyway, the pyruvic acid, I told you guys about those coenzymes, right? I'm going to repeat about the coenzymes first. We need to know something about these coenzymes.
Yeah.
So let me tell you about these. Remember I said that no amino acids are any good at carrying electrons, at taking electrons or giving up electrons.
They don't share. Gotcha.
Accepting or donating electrons. E minus electrons, right? And I mentioned this thing about these oxidation reduction pathways. Remember those? The oil rigs was the thing that was that mnemonic you go to remember. An oxidation reaction is the loss of a pair of electrons, and usually a pair of electrons, and often hydrogens, and reduction is a gain.
And does that lead to glycosis?
Say it again.
Oxidation and reduction that leads to the glycosis?
No. So in some of one, actually it's only one reaction in glycolysis, but a whole bunch of reactions in the Krebs cycle, a whole bunch of the reactions in the Krebs cycle, the oxidation reduction reactions occur. Okay. So I'm not going to worry about where they occur or anything else. That's not going to bother us. It's just I want you to realize, because this is important in understanding, see the electron here? That stands for the electron transport system. So that's why this electron thing is important. That's what I need to talk about. So no amino acids are good at accepting or donating electrons. So in oxidation, reduction reactions, coenzymes are used. These are the reactions in Krebs cycle. Coenzymes are used. And the main one, remember I mentioned this main one is NAD+. And NAD+ can accept 2 electrons, but it only accepts 1 hydrogen ion, and it becomes NADH.
No charge?
No charge.
All right.
There was a plus charge, it gained
You did-
another plus and two minuses.
This is okay.
Now,
Oh,
somebody I
yeah.
think asked me what NAD stood for, and I said it's not NADS, that's but nicotinamide adenine dinucleotide, and that's all we're going to say about it. We won't say it again.
Thanks.
But the point is that it's something we can't make. We can't make ourselves. We need to get it preformed in our diet. That goes with this last time. It's a vitamin. And so if we get it preformed in our diet, it's called a coenzyme. And all that means is that it works with the enzyme. In itself, this is not an enzyme itself. It works with an enzyme, and the enzyme needs it to work, and it needs the enzyme to help
Okay.
to be there. So all this means is it works with an enzyme.
So yeah, it's not. The name is misleading. It's not an
It's not an
enzyme.
enzyme.
Okay.
It's a friend. It's a friend of an enzyme.
Okay, gotcha.
Okay, and the enzyme has names, so you don't need to worry about it. Okay, so and it turns out this electron. Remember, I said another thing that hydrogen ions. If the hydrogen ion is given off, it can just flow into the cytoplasm and make the cytoplasm more acidic, right? But electrons can't just float away. They have to be handed from one molecule to another. Okay, so if an electron is given off, something has to take it. Does that make sense?
Mm-hmm.
And then if this guy's happy with it, okay. But if he's not, he's going to give it to somebody else. And that's what's going to happen in this whole electron transfer thing. Is the electron's going to be handed off to one person, and he's going to hand it off to another person. I know. And then he's going to hand it off to another person, and eventually it ends up in some final electron acceptor, which we'll talk about. So, this isn't the only. I noticed I said the main electron acceptor and donor is NAD. As in the form of NAD plus, it's an acceptor of electrons. When it's in the form of NADH, it can donate them. It'll just be a reverse reaction. It'll donate 2 electrons and 1 hydrogen ion. And it'll be back in the form of NAD plus again, ready to do it again. Alright, so oxidation and reduction reaction. We're not going to get into all the details of this, other than I mentioned, the loss of electrons and all that. But I did use the word, even when I was talking about fermentation, and I said that CO2 is the most oxidized form of carbon dioxide. So you're taking a sugar to begin with, glucose, you're taking glucose to begin with, and it's going to go through Krebs cycle, and then it's going to go through cellular respiration, and in the process, the sugar molecule is relatively what we call reduced. Not totally reduced, but it's relatively reduced. And the process is going to be totally oxidized to CO2 molecules. You can't get any more oxidized for the carbon than that. That's the most oxidized you can get. It's going to be totally oxidized to CO2. Okay, so let's just think about that. So we started off, so now this is a 3-carbon molecule. So I'm just going to put my 3 carbons. This is my 3-carbon pyruvic acid, right? I'm just looking at one of them. 2 of them. It happens for each one. But I'm just going to look at one. So pyruvic acid comes into this mitochondria, and I came down below that line, so that means I'm now in the mitochondria.
Mm-hmm.
And one of the first things that happens is that CO2 is removed. That's an oxidation reaction. We're not going to get into the details, but don't worry about it. But just keep in mind that is. Now, so that leaves us with a 2-carbon molecule. We're not even going to worry about all the names. We're not going to worry about all the carriers. There's a carrier called coenzyme A. We're not going to worry about all that. But anyway, you're left with a 2-carbon. But sitting in the mitochondrion is a particular 4-carbon molecule that's already there. It's already there, just sitting and waiting for its big moment. And its big moment is when this 2-carbon molecule shows up. Then it can get together with them, forming a 6-carbon molecule.
Yeah.
By the way, it's citric acid is what it is. But
yeah.
citric acid. But you don't need to worry about this.
But isn't the Krebs cycle like the citric cycle?
Yeah, that's why it's also called the citric acid cycle. Because the first thing that's formed is citric acid. But I'm not going to worry about it. But yeah, it can be called the Krebs cycle. I like to call it, this is the Krebs cycle.
And that's how we get our 6
I like
carbon
to call
sugars.
it the Krebs cycle because it gives credit to the guy who found out most of these reactions. Hans Krebs, I think his name is. It's also called the citric acid cycle. It's also called the TCA cycle, which stands for tricarboxylic acid cycle. So all kinds of fancy words and never mind. Krebs cycle.
That's where we get the 6 carbon sugars to be broken down in the.
Not actually. But anyway, and by the way, I call it the dreaded Krebs cycle. You've heard me say that before because people go, Krebs cycle, I never can understand it. I'm not having you memorize details about the Krebs cycle. I'm having you memorize overall process so you have an understanding of what's happening. So initially then we're entering this 3 carbon pyruvic acid. A CO2 gets removed, we're left with the 2 carbon molecule. It joins with a 4 carbon molecule that happens to be there, ready for its big moment. And then this 6 carbon molecule goes through a series of changes with another CO2 being removed. I should put the CO2s in different colors to see. Let's do that. Let's do CO2s in red. CO2. Here's a CO2. And I'm not worrying about where it happens or what, I'm just showing the basic idea. Well, now if I took a CO2 off of a 6 carbon molecule, how many carbons do I have left? There's 5, right? So there's now 5 carbons. And that goes through a series of changes and another CO2 is removed.
And then becomes 4 carbon.
And then so I'm left with a 4 carbon molecule now. And this 4 carbon molecule goes through a couple of changes and ends up back as that original 4 carbon molecule that you started from. Seems like magic, but it's not magic. It's just
No,
the
it's
way it
the
works.
cycle.
There is 1 ATP made. If you want to be fancy, we could have 1 ATP being made here. Because it does happen, but just 1.
What is the purpose of the CO2?
Well, that's going to be the waste product, right?
Okay, that's all it is.
But now, so now, we're not quite done with what happened in the Krebs cycle, but to begin with, if I said, what have we accomplished so far? We've started with a 3 carbon pyruvic acid, and so far what have we done? We've totally oxidized it to 3 CO2 molecules. 3 carbon pyruvic acid, 3 CO2. So we've gotten rid of all the carbons that we've entered. But I did say totally oxidized. And you see, let's just say that, I'll put it over here. So, in the Krebs cycle, and I said I'm not quite done with it, Krebs cycle, the 3 carbon, I mean, you want to be technical, pyruvic acid is not part of the Krebs cycle because we have to remove the CO2 first, but I'll still say it down here. The 3 carbon pyruvic acid is totally oxidized to 3CO2. That's it. Can't get any more oxidized for carbon than that. But oxidized. And if you go back to what I gave you the definition of oxidation, most biological oxidations involve the loss of a pair of electrons. We need something to take a hold of those electrons. And what are our electron acceptors and donors to begin with? NAD+. And when they accept the electrons and hydrogen ions, what does it become?
NADH.
So this is the oxidized form of the the NAD+. This is the reduced form.
And the 2 electrons transfer by ionic bond.
No. It's no, because the electrons are just. Oh God, how do we do this? Electrons. It has an. Let's just say that NAD+ has an affinity for electrons. That didn't tell you much, did it?
No.
But the electrons are handed to it, and It has other hydrogen and it rearranges the molecule to form an ADH.
I just thought it was because the oxygen was 6, the electrons.
No oxygen involved here.
No?
We're using the word oxidize, but that has to do with electrons.
Oh, okay.
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 We haven't touched the oxygen yet. Let's come in.
Losing, oxidation is losing.
Oxidation is a loss of a pair of electrons.
Then why is
So
it
the
adding?
carbon here is oxidized, so it's losing electrons, and the oxidized form of the NADH is accepting it, so it becomes reduced.
Okay, so that in NAD plus is oxidized form, and then you're adding the 2 electrons.
Yes, and so it's now reduced. Yeah.
Okay. All right.
See, and that I really, if you understand it, that's great. If you didn't quite understand it, it's okay because you'll still be able to understand what I'm trying to tell you. But I'm glad you asked and you did understand it. That's good. But so now let's finish it. So in the Krebs cycle, what have we done so far? We have completely oxidized that initial 3-carbon molecule to 3 carbon dioxides. And I'm just going to do like this. I'm not saying where they're coming from, and I'm not even going to say how many. Let's not worry about it. And we're making many, and again, we're not going to even worry about it, reduced coenzymes. Most of them are NADHs, but there's another one called FADH2 if you read about it, you see it. So in the Krebs cycle, the 3-carbon prolific acid is totally oxidized to 3 CO2 molecules with many reduced coenzymes formed. These are important molecules because these have now accepted these electrons, but they've accepted electrons, and they want to give them up to somebody else who'd rather take them from them. And that's this whole process called the electron transport system. So these now enter the ETS, or they go to the ETS, the electron transport system, and give their electrons and hydrogen. Okay, but let's stop for just a second. If you look at, I'm going to erase this. If you look at a mitochondria, singular mitochondria, it's a membrane-bound organelle, but actually has double membranes. Nuclei have double membranes too. That's just whatever. So if this is a mitochondria, there's an inner membrane like this. That wasn't very nice, but that was okay. So this is a mitochondria. So this is mitochondria, and so there's the outer membrane, and then there's the inner membrane.
Ooh. What's the membrane made as? Phospholipid?
Yep, phospholipids and proteins. Okay. Just for your knowledge, it's not going to appear on the test or anything on the next test, but what they call the electron transfer system, or the respiratory enzymes are often called, are embedded in that inner membrane, actually embedded in the membrane. All the Krebs cycle stuff, all the Krebs cycle reactions, they take place out here, which you could call the cytoplasm if you want. You could call it there's a term for it. It's not stroma. What is it? There's a term for the fluid in the mitochondria. I can't remember what the term is right now. And all those Krebs cycle reactions are just taking place in that. But the electron transport system, the ETS, all that is taking place in the inner membrane, in the membrane itself. Okay. In other words, the actual enzymes and everything of the ETS are embedded in that membrane. You need a membrane for a respiration, for a respiratory system. That's why I say that, people that, this is biochemistry. This is biochemistry of when I talk about bacteria or something. Most biologists don't study this stuff. But I did mention that there are bacteria that do anaerobic respiration. they have a set of respiratory enzymes embedded. They don't have mitochondria, but they're embedded in their cell membrane. And the aerobic bacteria, see, I mentioned that cellular respiration is in the mitochondria of eukaryotic cells. The bacteria that do it, the respiratory part, the electron transfer system, is embedded in their cell membrane. They don't have intermembrane, outer membrane, because they don't have, they don't have mitochondria, but they have a cell membrane. And they have the respiratory enzymes are embedded in their cell membrane. And those bacteria that do anaerobic respiration, they're embedded in their cell membrane. They just don't use oxygen. Okay, they use something else. But we haven't even talked about what oxygen's used for. We're getting there. We're almost there, gang. God, we're going to do it today. Yes, we are. Okay. So, so the ETS. So you make a whole bunch of these reduced coenzymes. And these reduced coenzymes, what are they? They're carriers of electrons and hydrogen ions. And they're going to enter the ETS and give those electrons and hydrogen ions to the electron transfer system. So the reduced coenzymes, and what's the main one? Mainly NADH. Give their E minus and H plus to the ETS, and the ETS stands for electron transport system. Electron, we're spelled it right. And it's actually, there is a chain of these molecules that do this, so it's often called the ETC for electron transport chain. Same thing. They're also called respiratory enzymes. Same thing. They all mean the same thing. Okay. And this electron transport system is embedded. I'll say which is embedded. For our purposes, it's not a big deal that it's embedded in the membrane. But I'm going to tell you that it actually is a big deal in terms of how it works, but we're not getting into that kind of stuff. Which is embedded in the inner membrane of the mitochondria. Okay. Now, what's the big deal here? So the NADH gives us these electrons and hydrogens to this system. And the electrons then are handed from one carrier to another, to another, to another. It eventually has to end up in some final acceptor of the electrons. But in the process of these electrons being passed from carrier to carrier, you can think of them as high energy electrons if you want to. In chemistry, we talk about reducing potential. But in the process of having these electrons transferred from one molecule to another, ATP molecules are made. Now, the method that is made is very tricky, very complicated. The guy who originally came up with the idea of how ATP is made, his name was Peter Mitchell back in 1961, a long time ago. But it took years before anyone even understood what he was talking about. He's a brilliant guy in England. And then they started doing experiments, and they go, hey, this seems like what he's talking about makes sense. And now they know that it all makes sense. It all has to do with, and you don't have to write any of this down, I'm just going to say it real quickly. But as these electrons and hydrogen are being passed from carrier to carrier, every so often some hydrogen ions are pumped out into this space in between the two membranes. And then they start building up out there, and they want to come back through that membrane, and there's certain channels that allow them to come back through. And as they come back through, they force the making of ATP. That's really tricky stuff, and we're not going to worry about it. It's called the chemiosmotic theory, and they talk about the electromotive force. That's all words. Doesn't mean anything for you guys. Don't worry about it. But let's go on. So the reduced coenzymes give their electrons and hydrogen to the electron transfer system. Continuing on with this, the electrons and H plus. And see, sometimes electrons are transferred with the H plus, sometimes just the electrons are transferred. So that's why I say whatever. But the electrons and hydrogens are transferred from one molecule to another. That's why it's called the electron transport system. And ATP is made in the process. Now, I'm not worrying about numbers, I'm just worrying about final numbers. Well, what did I say here? I said that for each pyruvic acid that comes in here, we make a bunch of coenzymes. If you want numbers, or NADHs, but don't worry about it. I'm just saying for each pyruvic acid that comes in, you get many NADHs made. Okay. For each NADH that goes, and gives its electron to the electron transfer system, you get 3 ATP. Okay, so 3 ATP made for each NADH that begins the ETS. And actually, those NADHs, it just gives electrons, then they go back and get some more electrons from the carbon cycle, go back up and get some, get some more. So it's just, the NADHs are just carrier molecules. That's what they are. They're just carriers. Carriers of electrons and hydrogens. And when they get some electrons from Krebs cycle, they'll go to BTS and give them off. They go back to the Krebs cycle, get them off, and they're all right here together. I mean, so Krebs cycle's all happening in here, and so these guys are just bouncing around doing this.
And the purpose of transferring the electrons is to make ATP.
ATP.
That's all I needed to know, really.
Yes. Now, those electrons, they're going from carrier to carrier, but you have to have some final acceptor of those electrons, some molecule that is the final acceptor of the electrons, and then you have a waste product.
Mm-hmm.
And guess what that final acceptor is? I'll give you a big hint.
Oxygen.
Oxygen. Okay, oxygen. So
That's what I said.
oxygen.
Takes two electrons.
Oxygen is the final acceptor of the E- and H+ electrons and hydrogen ions. I'm going to give you the formula, and it's going to look funny, and I'll tell you why. But let me write it down. So here's the formula. 1/2 of an O2 molecule plus 2 electrons plus 2 hydrogen ions give you. What do you think they're going to give you? Water. Water.
Yeah, CO2. Yeah, because that's why it's
CO2,
a
H2O.
or H2O.
H2O. So in other words, the oxygen you breathe in ends up in water molecules. Okay? That makes sense?
Yes. That's why they say it's H or O1 half O.
Well, see, here's the deal. The reason you
H
write
half O.
it's funny you say half of an O2. Why don't you just write O? And it's weird. Oxygen never appears as a single atom.
Atoms.
In nature. It's always O2.
O2.
It's always an O2 molecule.
I get it now.
What? The single.
We don't personally make water.
Yes,
Yes, we do.
Yes, no, we're constantly making water. Every time you are running
Yeah,
these
we do.
reactions, you are making water. That's called metabolic water.
He just said.
And if you look in a physiology book, they'll say that in any given day you'll make a certain number of mils of metabolic. Because they talk about your water, you drink a certain amount of water. But when they talk about metabolic water, that's water that is made in various kinds of reactions. This is just one reaction where we make water. Okay.
Awesome.
And so, but yeah, no, it's actually making water. And this oxygen.
No longer has the two. I get
This
it now.
oxygen is what you breathed in. It was an O2 that you breathed in originally, and it ends up in a water molecule.
That's
Yeah,
awesome.
oxygen, you breathe it in. But see, you never get oxygen as a single atom, a singlet oxygen.
Because
So
of
that's
theirs.
why they write as 1/2 of an O2, because they don't just say, otherwise it looks like you just take an oxygen. It's a little tricky the way it actually works. But the enzymes do it. They can handle it. They can take an oxygen molecule
And
and
divide
then
it.
they can hook on one of them, hook 2 electrons and 2 hydrogens, and on another one, hook another 2 electrons and 2 hydrogens, and then it'll come off as 2 waters, you see. But that's why we write it that way. So it looks funny. But so, let me answer your question. Let me just summarize all this. Yeah.
So that all is cellular respiration.
Yes. So officially, then, cellular respiration involves the Krebs cycle and electron transport. Okay. So cellular respiration involves Krebs cycle and respiration. And if you didn't have oxygen, then you wouldn't have the spinal acceptor, and pretty soon things would just start backing up, and you couldn't run it anymore. That mitochondria wouldn't do anything. So you need oxygen to make all this work.
That's cool.
So let's do it. So I want you to try to get as much understanding of this as you can. Let's just go back again real quickly. So what if you ask me? Yeah, go ahead.
So this whole cycle, is this all part of the Krebs cycle? Is it all?
Well, remember we did start with glycolysis up here, didn't we?
Glycolysis, Krebs cycle.
But all that did is break down your a sugar into 2 pyruvic acids. Once we got that, and if you're aerobic, you have mitochondria, those pyruvic acids go into the mitochondria to do the Krebs and electron transport. And of course, why is that so useful? Because look, 34 more ATP than we got from just the glycolysis, or if you were
just from breaking down sugar.
2 for every sugar. You only get 2 glucose, 2
ATP.
ATPs for every glucose. If you use glycolysis and then Krebs and ETS, we'll get 36 total ATP for every glucose. So much more trapped energy or available energy, or however you want to put it, whatever word you want to use. Yeah.
So only the reduced coenzymes go to the ETS?
Correct.
What happens to the 3CO2?
Yeah, I mean, this is coming out.
It's carbon dioxide.
This comes out. It goes. It comes out of the mitochondria into the cell. From the cell to the blood. From the
Blood to the capillaries. Capillaries released. Gotcha.
So, so this is. I mean, you can see just how valuable this is in terms of trapping energy. There's a lot of available energy in glucose, but we want it in a usable form, and our usable form is ATP. And and the numbers they come up with, and I don't know exactly what they are, but of trapping the amount of usable energy that we trap for the breakdown of glucose. Remember, we always lose the second law of thermodynamics. We're going to lose a certain amount of energy in the form of heat and non-usable energy for our purposes. But the ATP is usable energy, so we trap some of the available energy as ATP. And it's something like 40% efficient. I don't know the exact number, but somewhere around 40% of the available energy in glucose is trapped in ATP, which is more efficient than an automobile. Automobiles, I don't know, 20-some odd percent or something like that. Diesels are a little more efficient in terms of using the available energy in gasoline. So it's just in terms of efficiency is pretty good. It's not super great, but it's pretty good. Yeah.
I just want to make sure I'm clear on the end result. Things will start as glycosis, and eventually it will end up as H2O.
What?
Like the
So.
It's the final product of all.
Okay,
of
so
H2O.
the final. Okay, so what is the final? And I'm going to write this down as an equation. But what is the final product then of the complete breakdown of glucose?
CO2.
Complete breakdown of glucose
ATP
would be a bunch of ATP, CO2, and water.
And water.
And I'm going to give you the formula right now. Okay, I was just going to do that. So this is good. So this is the formula, and it's not like you need to memorize the formula, but I think maybe you should.
I'll do it. I'll do it
Here's
right
the
now.
formula. C6H12O6. This is glucose.
That's easy.
And you use 6 for each glucose. You use 6 oxygen molecules, and you get out of it 6CO2 plus 6H2O plus, I ran out of room here, plus 36 ATP.
Yeah.
Okay. So And if you actually count up all these things, you'll see that it's 6 carbons, 6 CO2s. You know, count up all those things.
36
Point
ATP.
is,
Got it.
so this would be the overall process then of the complete oxidation of glucose. Starting with glycolysis and then using respiration, gravity, ETS, is CO2 as a waste product, H2O as a waste product, and ATP. Inorganic. CO2 and H2O are inorganic, right? No organic molecules left over as there is in the fermentation. Totally inorganic, but very efficient. Very efficient. Okay, so let's just, I want to restate it one more time. I'm getting too bored. Just take a little nap. But just to make sure.
Trying to prevent that.
Most organisms on Earth start with glycolysis and break it down to pyruvic acid. If you are aerobic, those pyruvic acids will go into the mitochondria, and in the process of going through the Krebs and the electron transport system, 3 carbons of the pyruvic acid are totally oxidized to 3 CO2s. You make a whole bunch of reduced coenzymes, which feed into the electron transport system, and then you make a whole bunch of ATPs. 3 ATPs for every NADH. And then what's the final acceptor of the electrons?
Oxygen.
Oxygen. To make as a waste product?
Water.
Water. That's a nice waste product. It's not toxic or anything, is it? It's a nice waste product.
No,
it's a good
good
one.
stuff.
CO2 we breathe out easily. It's easy. We just breathe it out. So it's an easy thing. If you're a yeast and you're making wine, for example, now the yeast doesn't know he's making wine. He's just fermenting the sugar. He wants to make more yeast and he's growing, dividing, but in the process he's using up sugar. But what is he making? He's making alcohol. Well, alcohol is a waste product, and it becomes toxic if it gets too high, and it gets up to about 11 to 12%, and then those little yeasties can't keep growing. They stop growing because they're sitting in their own waste product. So, the nice thing about our waste product, water, is not a bad deal for us, and CO2 we just breathe out. Yeah, let me start here first.
So, is that equation the equation for cellular respiration?
I would just say it's for the complete oxidation of sugar, which includes, in this case, glycolysis, and cellular respiration. Or you could just say complete respiration, whatever you want.
It's the Krebs cycle.
So once glucose is totally broken down,
and you get the waste product being the H2O. So does the
And
H2O go to
CO2.
plasma? Is that
I mean, it's just
That's my
it's
problem.
part of the cell. If there's too much water in the cell, it'll go out of the cell.
So it ends up being like the plasma.
Sure. I mean, if you're just a single cell, it's just part of your cell, or you can diffuse out.
Yeah, you got that.
Osmose out.
Yeah.
Okay.
So once each thing is made, it kind of goes and does its own thing. Like the CO2 will go do its thing.
Well, CO2 just, it's just a gas. It just diffuses out of the mitochondria. And the ATP can diffuse out and be used by the cell to do, maybe make some protein.
And it's diffused into the blood, though, right?
Well, if we're like humans, then what? The CO2 gets in the blood, not the ATP, of course. That stays in the cell. But,
Does this exchange happen in the lungs?
For the CO2, sure.
Well,
to get rid of the CO2.
Once blood hits the blue blood hits capillaries, it has a process where it releases off the CO2, takes an oxygen.
So it's easy. It's a nice, easy waste product because, if it built up in your system, then of course you would get too acidic. But we get rid of it. So. Question? Question.
Do they ferment like alcohol with higher alcohol content?
Oh, you mean in other words, how do you make things with higher alcohol? You have to. Right, you have to distill it, which means you heat it. The alcohol will burn off faster than the lower temperatures
and
of the
then
water. So they get the alcohol coming off as a gas, and then they'll cool it off somewhere over here to collect it. So now they have pure alcohol, and then they'll take that pure alcohol to some other, like some wine, and add it to the wine to make, say, cognac or something like that.
Oh yeah.
And then they do the same kind of thing for making.
Brandy.
To make things like whiskeys and stuff like that, they're not using grapes. They're using corn and I don't know what, rye and all those other things that are really used. Wait, there's another question. Yeah.
When larger organisms produce that excess water, doesn't in fact find its way out through urine
Oh yeah, of course. I mean, that's the
Yeah.
thing. I mean, or sweater. Sure. If you're a plant, you can come off as transpiration.
Yeah.
But that's why I say it's a nice waste product because it's not toxic.
Right, but if we kept on, but it's
Oh yeah.
hypertonic situation.
Well, we'd start to blow it or something.
Yeah.
But no, we'll get rid of it. Now, okay. Now listen. Here's the deal. One last little bit. And I didn't think I'd get to photosynthesis. I was hoping to.
Oh, wow.
That'll be, well, the test is Wednesday, so that'll be next Monday. And we'll cover photosynthesis in a very short, quick way because I don't want to get too far behind here. But there is still one last thing about all this. I did say that the cellular respiration starts with the glycolysis, and I said, or fatty acid metabolism. So let me quickly tell you about that, because this is sort of interesting. And this is why, fats are a nice, easy thing to store, and they're great to use for energy, and I'll show you why.
I wanted to know, what are fatty acids made out of?
Well, I'm going to show you that.
Okay.
So with fatty acid metabolism, we don't need to worry about glycolysis. There's no glycolysis involved with fatty acid metabolism. And fatty acid metabolism all happens right there in the mitochondria.
Okay.
Fatty acids go into there. So let's do this. So this is kind of neat, and you'll see that just a little bit there. So fatty acids. Fatty acids are long chains of carbons and hydrogens. Carbons and hydrogens. Remember I showed you that carbon. I just said Google it, carbon.
Little tails come off of it.
Hydrogens in each of these places here, and I'm not going to write them all in.
Yeah.
Boom, 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 and there's no HT. Don't worry about that. But what happens in fatty acid metabolism? Now notice over here, what was this thing that we had? I just erased it, but it was a 2-carbon molecule right here. Remember, that was a 3-carbon pyruvic acid and 1 carbon dioxide.
Came off,
One
yeah.
carbon kicked off as carbon dioxide. We're left with 2 carbons. Aha! What happens with fatty acid metabolism is we take 2 carbons at a time, and they immediately feed directly to the Krebs cycle.
Oh.
No glycolysis.
Oh.
And then the next 2 carbons feed into it. Then the next 2 carbons feed into it. So it's very quick. So I'll just make it simple. In fatty acid, I'm going to put Fa period for fatty acid. Fatty acid metabolism. It occurs in the mitochondria. That's where it happens. Fatty acid metabolism. 2 carbons are broken off at a time and feed directly into Krebs cycle.
So utilizing the fatty acid gives you, wow, okay,
So we don't use, so no glycolysis is involved in fatty acid metabolism. So in fatty acid metabolism, 2 carbons are broken off at a time, broken off of the fatty acid, and feed directly into the Krebs cycle.
So there's only 2 CO2 put off then?
Say it one more time.
There's only 2 CO2 put off in fatty acid?
Yeah, per 2 carbons. Yes, exactly. Feed directly into the Krebs.
So when it's done with fatty acids as opposed to
Say it one more time now.
when cellular respiration is happening with fatty acids instead of the glucose, does it only make 34 ATP?
No. We're going to get more. Actually, it turns out we're going to get more. But don't worry about the details. But just the reason why it's a good deal is that, and I'll put this, now how many ATP did we get for a 6-carbon sugar?
36.
36 total. If I take 6 carbons worth of fatty acid, just to compare the two.
Oh,
carbons,
right. You get
okay,
72,
wait a minute, for
108.
every 6 carbons of fatty acid, 44 ATP.
Oh, well, then that's even.
So it's a little more efficient in terms of overall ATP production per carbon. So, if you're comparing 6 carbons sugar with 6 carbons fatty acid, we get 44 ATP if we break down totally oxidize that fatty acid versus 36 if we totally oxidize the glucose. We'll still get CO2 and water as our waste products. Okay. So we'll still get the same number of CO2 and H2O, but we get more ATP out of it. So that's from your body's perspective. That's why fat's such a good thing. It's easy to store and it's a great energy source because we actually have more. We can make more ATP when we burn it. Metabolism. Does that make sense? What I'm saying?
Oh yeah.
There's kind of a lot. And don't worry
Explains
about
why
the
you get
detail.
tired
I'm
a
not
lot.
going into the detail. I just want you to realize that in fatty acid metabolism, let's write this. In fatty acid metabolism, no glycolysis. So it's a little more efficient because remember, it's the Krebs and ETS that allow you to make so many ATP. So it's much more efficient. So no glycolysis that occurs totally in the mitochondria, and you actually end up with more ATP per carbon or for 6 carbon. I gave you for 6 carbon, but that makes sense. So it's a pretty neat deal. Okay, we have 5 minutes. I'm not going to start on photosynthesis. 5 minutes to ask me any questions about the exam.
I just am kind of curious. I mean, I'll figure out the math of it later. Why that's 36 and why that's 44.
Because we'd have to go through and how many reduced enzymes. Don't worry. Say it
44.
It's 44, not
44.
44 total per 6 carbons. Yeah. So I'm 13 questions and most of them are like discussion. I'm on the final, right? On the
Midterm, yeah.
Well, I'll ask you a question about the differences between prokaryotic and eukaryotic cells. Some of the differences. I'll ask you about covalent ionic bonds. I'll ask you what are those? I'll ask you about the macromolecules. I'll ask you about fermentation. I'll ask you about metabolism, catabolic and anabolic reactions, and what's ATP have to do with those guys. Remember that? Catabolic reactions, you're breaking things down, you're making ATP. Anabolic reactions, you're putting it together and you need ATP to make them work.
So you use the energy from the high energy.
And I'm not expecting super detail on this. If you just can give me those ideas. And don't leave anything blank on the exam. If you don't leave anything blank, because I can't give you any credit for blank, and if you give me something, I can give you some partial credit. Before you go, if there's anyone that needs, I have a few names already written down, but if there's anyone who needs a test in the testing center, did I put that up there? Oh yeah, I did that. So be sure to tell me so I can put the test in the testing center for you.
Why would you need, in case you can't be here, or is that?
Or you need extra time or something.
Okay. Oh yeah, I have that, but I deal with it anyway. Oh come on, just turn off, would you? You always do this to me.